First lesson, nutrition in plants. So the plants can synthesize food in the manner of autotrophic nutrition. Autotrophic nutrition. Okay, it means in the process of photosynthesis, it can synthesize their own food. That type of nutrition is known as a autotrophic nutrition. So, which part of the plant can produce food? Very good. Leaves can produce food material in the process of photosynthesis. So, photosynthesis is the source of metabolic activity to produce food material. That food material is consumed by every organism which are living on the earth. Okay? It means all the organisms directly or indirectly depend upon the photosynthesis. That's why plants are called as a universal food providers. Okay? So now we are going to know about the leaves are producing or the leaves are performing photosynthesis. Okay? So you can prove it with the help of iodine test. Iodine is a special character which can turn into blue-black color when the presence of starch in the leaf or any substance. Okay, so that's why we are using iodine solution to prove food material. Starch is present in the in the leaf. So leaf is the part which is the green leaf is the part which is producing food material or starch or carbohydrates. Is it right or wrong? Yes. So, aim is to prove that presence of starch in leaves. It means if there is a presence of starch, the starch is prepared in the process of photosynthesis. Okay, you can easily denote it. It means you can easily say it, right? So required materials. Beaker and test tube. Beaker, test tube, boiling test tube, tripod stand, Bunsen burner, petri dish, and healthy green leaf. So you can take it as a material also. Okay. So operators are beaker, test tube, boiling tube. Test tube or boiling tube, tripod stand, Bunsen burner, petri dish, and petri dish. So healthy leaf is considered material. Okay. Next one, chemicals. Which type of chemicals we are using here? One is methylated spirit or ethanol. Okay. Methylated spirit. So and iodine solution. So water. These are the chemicals we are using in the experiment. Okay, so Advaita, yes, so next chemical procedure. So a leaf is taken from the potted plant. So it must be healthy leaf. Okay, so you can take the healthy leaf from the potted plant and the leaf is put into a test tube containing methylated spirit. We can take a test tube and we can pour some methylated spirit and then we can put that leaf inside of the test tube in spirit. So next, the leaf is boiled. Now we will keep this test tube in the water containing beaker. So now then we can flame the Bunsen burner and we will boil the beaker along with the test tube containing leaf. Are you understand? So that's it. It becomes when you boil the water, the methylated spirit containing or test tube containing leaf will get into palm color. Pale color. Okay, it means the green pigment present inside of the leaf will denatures. Okay. Why we cannot take leaf for the starch identification, carbohydrate or starch identification means we cannot get that much of good result. We cannot, the blue-black color cannot visible in green color. That's why if you turn it into pearl color, when you pour that iodine drops on the surface of leaf, you will get a prompt or correct blue black color, visible blue black color spots on the surface of leaf. That's why we want to boil the leaf. Okay, so now become pearl. The leaf is taken out of the boiling test tube. Now we can separate the test tube and we will take that leaf, pearl color leaf, and we will put it in the glass, watch glass. Okay, watch glass. So next, a leaf is taken and the watch glass or petri dish. You can use it. Okay. So next, a few, now we can pour or we can a few drops of tincture iodine. Tincture iodine or the betridine. We can use it for the iodine test. It means starch identification. So solution are added to observe the leaf. Okay. So now observation. When you add the iodine solution to on the surface of the leaf, so the iodine solution can reacts with the starch present in the leaf, and immediately it gives a blue black color. It indicates the blue black color indicates the presence of starch in the leaf. It means there is a process, photosynthesis. Due to the photosynthesis process, starch was prepared. We can prove in this manner. Are you understand?
Yes, sir.
Okay?
Yes, sir.
So, okay. Once again, see this experiment. The leaves can produce starch in the process of photosynthesis. In the process of photosynthesis. So, it means photosynthesis is the source for food material in the earth, on earth. Okay, so every organism directly or indirectly depends upon the food of the plants. It means that food material was prepared by photosynthesis process. Okay, so which part is responsible for production of food material in plant?
Leaves.
Leaves are the parts preparing food material through the photosynthesis process. Okay, so now if you want to prove that photosynthesis happens in a leaf, so you must do the experiment iodine test. Test. So for that you aim to prove that presence of starch in leaves. To prove that presence of starch in leaves. So that's most important. We are going to perform experiment with the help of healthy leaves. Okay. If you can observe any starch deposition in a leaf, it means that is prepared by photosynthesis process. Okay. Why you are taking iodine solution here? Iodine can react with the starch or carbohydrates and it gives blue-black color, prominent blue-black color. Okay, for this purpose we are taking iodine solution here. Okay, so aim is to prove that presence of starch in leaf. Required materials. So required operator solution. So beaker, test tube or boiling tube, tripod and Bunsen burner, petri dish, and healthy green leaf. These are the operators materials we need in the experiment. Next one, chemicals required. So are reagents. Chemicals are reagents required here, that is methylated spirit and iodine solution. Then water also we need. Okay. So next one, procedure. A leaf taken from the potted plant. We already discussed. So we need a healthy leaf to prove the presence of starch. That's why we are taking healthy leaf from the potted plant. Next one, the leaf is put into the test tube containing methylated spirit. So we will take one test tube and we can pour some amount of methylate spirit in it, and then we can keep that leaf inside of the test tube. That test tube we will keep it in the beaker of the water containing beaker. Okay, where which is shown in the figure. Okay, now we taken beaker and we taken water in it, and then we kept a test tube in it, which is containing methylated state and leaf. Okay, now we can use tripod stand, and we can keep that above the tripod stand we can keep asbestos plate. On that asbestos plate we can keep this beaker. Okay, then we can put a test tube in it. Then we can, with the help of Bunsen burner, we will boil the entire apparatus or entire arrangement. Okay, after boiling the beaker, we will get pale color leaf in that methylated spirit of the test tube. Okay, why? Because we are taking pale color leaf. Why we need it? Because for good result. If you perform the iodine test on green leaves, you cannot take that much of visible blue black color. Okay, so if you turn it as a pale color, you will get prompt and visible result. That's why we will boil that leaf and we will turn it as into pale color. Okay, so then after getting a pale color leaf, so we will collect that pale color leaf and we will keep it in a petri dish or watch glass. Okay, after keeping it in the petri dish, we can pour some drops of iodine solution plus betadine, or tincture iodine. We can pour or we can lay on the surface of leaf. Now observation. After pouring or leaving that iodine drops on the surface of the leaf, we will get blue black color spots on the leaf. It means, so there is a presence of starch. The starch was prepared in the photosynthesis. Easily we can prove it. Okay, result is the leaf contains starch solution. We proved with the help of this experiment. Iodine test. Iodine
Yes.
Are you understand?
Yes, sir.
Any doubts?
No, sir.
We can observe a blue-black color spots on the surface of the leaf or leaf. It indicates the presence of starch in the leaf, which is produced in the photosynthesis. Are you understand?
Yes, sir.
So this is the leaf which is containing blue-black color. This experiment proved with the help of iodine. So that's why it is called as an iodine test. Okay, iodine.
Yes.
Okay. I understand.
Yes, sir.